Next up is Linnell Williams. After that is Tyler DeWitt. Tyler DeWitt, 100,000 people watch his videos every day. He has 1 million followers and has just passed the 100 million mark for his videos, and he's so excited to share with you. But right now, you're going to hear from Linnell Williams. She's a PhD student in physics at Harvard. She is the founder of Women Plus Color Project. She got her master's at Fisk University, one of the historic Black universities. She studied physics at Wesleyan University, one of the top research undergraduate, small liberal arts schools. And she's studying physics, and I caught up with her not so long ago when she was working on viruses. So virus to me sounds like biology. You know, what's a physicist? You know, someone in physics doing with biology, she'll tell us a little bit about that. And the other remarkable thing is her high school, she didn't have physics or calculus as an offering where she was growing up. And it was someone who said to her, hey, you have some real skills. Why don't you do some engineering that got her going in this direction. She's a force of nature. We're really lucky to have her. And she's modeling the best of what I think the next generation of science leaders need to be. Ladies and gentlemen, I think my favorite person on this science group, Linnell Williams.
Oh, I started talking. Thank you so much, John. Let me share my screen quickly. Let's see. Wait. So while I'm setting up, oh, I'm not unmuted. While I'm setting up, I guess I'll tell you guys a little bit about myself. As John said, I'm currently a PhD student at Harvard University, and I'm originally from Memphis, Tennessee. And probably about 13 years ago, I was in you guys' exact position. So when I was growing up, I actually wanted to be a journalist. I had no interest in science. I was only introduced to science because I did pretty well in most of my courses during high school in science, but I really, really wanted to be a journalist. And I also really hated science fairs. Does everyone see my screen? Okay, great. And everyone can hear me. Great. Yeah, so when I was in 9th grade, I had no clue I wanted to be a scientist. I didn't like science fairs that much either. I thought they were pretty boring, but I think that part of that is because you don't really get to learn what scientists actually get to do when you're in high school. You get to take these courses, you take your physics courses, and physics is just physics. You take your biology courses, and biology is biology. You take your chemistry courses or whatever, and chemistry is just chemistry. When at the end of the day, we all end up eventually just becoming scientists. So today, hopefully, I get to talk to you guys a little bit about, and I think what the other speaker said earlier was, speakers were saying earlier about being passionate about something is really important. But I wanted to talk to you guys about a little bit about what I'm passionate about researching and why someone like me who comes from a physics background finds it really important to really be thinking about problems that we can solve from a physicist's perspective in the sort of biological realm. To kind of further talk about that, I specifically study virus self-assembly in particular. In short, I study virus physics, and I'm going to talk to you guys a little bit today about what virus physics actually is and why it might be important, especially for vaccine development, engineering, etc. But we all don't necessarily live in silos, and one thing that you learn when you get into undergrad and go into graduate school and you find that thing you're passionate about is science isn't necessarily as separated as you might think it is, especially at your level. You get to do a bunch of really, really cool things, and you truly can explore what you want to explore. So prior to the pandemic, we all knew what viruses were, but really in the past year, there's been a huge increase in the amount of research that's geared towards virus research, specifically looking into COVID-19. When we talked about viruses before, we talked about them with respect to how scary they were. And of course, they make people sick and they take people down. And things like HIV and Ebola, et cetera, are sort of trigger words for us when we think about virus research. But the larger field of virology not only focuses on vaccine development, not only focuses on how to get rid of viruses. Some of us are interested in how viruses work and thinking about their structures and thinking about how we can actually better understand them. And so since we've had this spike in this sort of interest in scientific research with respect to viruses, um, again, lots of labs, lots of folks in different industries. I've even seen papers from folks who were on the sort of high energy physics string theory side, publishing papers on like the Fermi Dirac whatever of COVID-19, because we so much attention has been given to the specific topic. But this isn't the first time that physicists have actually been interested in studying viruses. Since the discovery of the DNA back in the 1950s and 60s, there have been physicists and biologists who have worked together to come to think about these different topics, to think about what the structure of viruses are, why they exist, how we can sort of better understand them. But what is a virus? So the textbook definition tells us that it's a molecular structure that has the ability to infect living living host, essentially. So us, our cells, animals, etc. And a very simple virus is made up of two basic things: genome, that's RNA or DNA, and some of you guys might have taken biology, and proteins. And those proteins essentially protect that genome, or protect the RNA and DNA that the virus is made of. In my lab in particular, we're very, very interested in simple viruses. And the simple virus that we want to study is an RNA virus. So that means the genome that the proteins begin to protect is RNA. And we specifically study what is known as the bacteriophage MS2. Bacteriophage MS2 is actually found in our gut. It's made up of 180 proteins that protect its RNA. If you compare bacteriophage MS2 to COVID-19, it's simple in comparison. So COVID-19, we've seen, is also an RNA virus. It's protected by three kinds of proteins: the spike proteins, the envelope protein, these large spike proteins, the envelope protein, and the protein membrane. And the MS2 virus, the virus that we're looking at, is even more simple than that. It's just the genome is protected by one single protein. And that makes it an ideal system for us to study. Typically physicists like to stick with something that's very simple, something that we can work with, something that we can like sort of apply math to and expand on. And it's best to start small before we start going into more complex systems like COVID-19. So why is this really, really interesting, right? So these simple viruses, as we know, infect cells. And we think that the simple viruses might help us better understand what we call self-assembly, during the infection process. So when viruses infect their host, essentially it gets its RNA into the host. Afterwards, that bit of RNA that has been injected into the host is replicated, and all of those bits and pieces of like RNA are now floating around in the cell, and we have pieces of RNA and coprotein that are floating around in the cell, and some of those are turned into coprotein. And now all those random bits and pieces of RNA and proteins that are floating around, if you can kind of picture that in your head, just suddenly zip up and assemble into new infectious virus. And eventually they burst out of the cell. What I want to understand is specifically the process of how these random bits and pieces of proteins and RNA begin to assemble into these structures. And we believe that self-assembly is a vital part of the RNA virus life cycle. So the reason why this is important is because we think this is interesting, and we believe this is self-assembly, is because not only does this happen inside of the cell, it happens outside of the cell too. If you take random bits and pieces of RNA, viral RNA, mix them with proteins, you end up getting viruses, and you can check these with TEM. And that tells us something about the physics that might be happening within the system, right? So because this happens inside of the cell and outside of the cell, and you compare images of in vitro assembly and in vivo assembly. In vivo means wild type, so viruses that are that come from the host. In vitro means that we take random bits and pieces of protein, RNA, mix them. You end up getting very similar structures, right? And this is why. This is where we began to think about the physics, right? So I don't know if you guys have taken chemistry yet, or talked about thermodynamics or statistical physics just yet in your career, but because this happens, we believe that this is a random process. we believe that the way the virus is able to allow its proteins or get the random bits and pieces of protein to assemble around that strand of RNA is by doing something called minimizing the free energy of the system. And to minimize the free energy of the system, all that means is um, all that tells you is the amount of work it takes for me to take those bits and pieces of protein and sort of stick them around and protect the RNA, right? And so we can kind of, think about this on a map and say that this essentially means that um, for the virus to go from random bits and pieces of RNA and proteins to a actual stable structure, it has to explore these different states. So sometimes it's just a bunch of random bits and pieces of protein and RNA, and other times it can probably assemble into other structures, such as a full capsid, or a capsid that's completely completed itself, a partial capsid, or even a monster capsid or a malformed capsid that might represent the system that we have. And because of this, the question that I want to answer and the question that I'm very interested in is thinking about, how viruses manage to assemble into their stable structures or assemble into a full virus, and why does it sometimes, also form other structures that might be weird. And to do this, what I want to do is create a map, essentially. We call that a phase diagram, right? And to do our experiments, we essentially do, as I said earlier, we take RNA, we add protein, and we watch it assemble. And then we create and we measure that. And we create a map that essentially represents, the areas, the sort of conditions that allow us to see full capsids, full infectious capsids, areas where we might see partial capsids or weirdly formed capsids, essentially, and areas where we might even see monster capsids that might form. And to do that, we have to sort of tweak the environment so I'm kind of walking you through my scientific process for this. That's the question that I want to answer. And so we take advantage of the tools that are available in physics and biology to answer those questions. So we can do things like change the environment or change the buffer, or change how much salt is around the virus, or how many proteins are in the sample, or how much RNA is in the sample. And then we begin to measure that. And we measure it using, two tools that are common in physics and biology, and that's gel electrophoresis and dynamic light scattering. Gel electrophoresis essentially tells us how big the stuff in our sample is. And so we can use these gels to essentially measure whether or not we have medium-sized items that look like the virus, smaller structures, or large structures that might also look like the virus. And then we also use a technique that called dynamic light scattering, where we shine a laser onto our sample and we measure the light that's being scattered off of it. And we take advantage of some physics. So Einstein introduced us to diffusion and Brownian motion. You can ask me about that. So we introduce some physics into this, and we take advantage of physics and say, okay, if the light fluctuations are really small, then the stuff is small. If it's, bigger, then we might see a virus. If it's really, really big, then we might see a monster capsid. And we use these different techniques to begin to build this map, essentially, that I want to build to better understand how viruses self-assemble. So why is this important? It's important because if we can understand the way that viruses assemble themselves inside of the capsid or outside of the capsid, we might be able to think about how viruses disassemble themselves. And then we might further think about how to control that process, right? And that might lead to new, more innovative ways of thinking about, vaccine development or managing viruses or phage therapy and taking advantage of what viruses have to offer. If you have any questions, just send them in the chat. But thank you so much. And here are my two cats as well. Samson and Delilah, that's their names. But thank you.
So, ladies and gentlemen, that was Lynnell Williams. And, if you want to be a PhD student, you can kind of get a sense of what one is navigating right now. Lynnell, what do you see for the future of your career? Like, what's the next few years look like? And maybe talk a little bit about what it's like being a woman and a woman of color in the science space.
Yeah, I'll answer the first. I have finally accepted that I like academia, despite all of the tweaks, all of the things that come with it. I do ultimately want to be a professor. So I am in my fourth year preparing for that and trying to wrap up projects and write papers so I can think about postdocs as my next step. As a woman of color in STEM in particular, I think I've had to navigate the sort of experience of being both a woman and also a person of color. And that also comes with its challenges and advantages as well. I'm also a low income student. And I think
That was
that,
my follow-up question for you to comment on that too.
yeah. So I think that, I have had those negative experiences as a result of my gender and my um, and my race. I think it was in undergrad, someone once told me, oh, yeah, I think they couldn't believe I was in the program. So they just assumed, oh, you must be one of those smart Black girls. Or I think when I first got to Harvard, it was assumed that it was told to my face essentially that I only got in because I was a Black woman. And I don't think people realize how hurtful those comments are. And I just kind of politely said to my colleagues, I'd match my resume and my scores with you any day. um, that I'm here because I'm actually good. And if it was just because it was a Black woman, there'd probably be a lot more of us sitting in this space. Um, just to give you a sample, but I think that I do cherish the fact that I do come from a different background and I'm able to inspire not just other women of color, other Black folks, other women. I'm also sort of breaking barriers for what a scientist looks like for non-Black folks or for folks who are not women. Part of the reason why it's so hard for people to encounter me at my stage is because they're not used to seeing people that look like me do science. And so it's important for me to be present, even, not just at events where I'm inspiring women of color in particular, but also other events so that we can ideas being scientists.
And you're the third woman of color PhD in physics at an Ivy League school, is that right?
So I'm the third Black woman to be admitted into the physics PhD program at Harvard. I was
Yeah.
the second Black woman to graduate from Wesleyan
Great.
when I was there, and you get used to those numbers, seconds and thirds. But eventually, I would love to see that we're not always talking about first and seconds and thirds. Um, Maybe 10 years from now, hopefully. Um,
Great.
and
Well,
And
thank you.
and
If people want to follow up with
and I
her.
think we have a question. No, I think we have a question for you from one of our fabulous students, Daniela. Daniela, come on, can you please unmute and fire away, please?
Oh, I was wondering if, like, once the virus has been formed, could you break the protein shell? And if you could, what would happen?
That's really hard. My colleague is actually trying to answer that question. So the thing is, if you saw that picture of the well, the really, really deep well, right? That represents the energy it takes, right, to get to that well, essentially. The deeper you are in the well, the harder it is to get out of it, right? It's really, really hard. So you can't necessarily it's hard to disassemble it the same way. It's hard to pull it apart the same way you put it together, essentially. And folks have been trying to figure out how to disassemble systems, um, and make it in a sort of reversible way for a very long time. Yeah.
Great. So thank you. So for time, we got to move on.